Hello, this is Donna, Lauren, and Chelsea. We experimented the effects of zinc 2 plus concentrations on enzyme activity. Catalase is an enzyme that converts H2O2 into oxygen. H2O2 is damaging to cells, so it is important to measure the enzyme activity. It is also important to test different conditions and the effects of these on the enzyme. In the Dong et al. Study, the addition of zinc 2 plus inhibited the enzyme activity. Because of this added zinc, the productivity decreased by 60%, which is shown in the table. We studied the effects of different zinc 2 plus concentrations on catalase activity. Our hypothesis was, as the concentration of zinc 2 plus increased, the production of oxygen would decrease, similar to the results in the Dong et al. Study. We used 0.5 millimolar zinc chloride, 1.0 millimolar zinc chloride, and 1.5 millimolar zinc chloride concentrations, based on the concentrations in the Dong et al. Study used in their experiment.
For this experiment, we started by setting the spectrophotometer to 0% transmittance. Then, in a cuvette tube, we added KPO4 buffer, guaiacol, turnip extract, and hydrogen peroxide. We immediately placed the tube in the spectrophotometer and measured its absorbance every 20 seconds for 2 minutes. This was our control vial, and we repeated this for 2 more trials. Our second, third, and fourth treatments contained the same amount of KPO4, guaiacol, turnip extract, and hydrogen peroxide. However, the second treatment contained 0.5 millimolar zinc chloride, the third had 1.0 millimolar zinc chloride, and the fourth had 1.5 millimolar zinc chloride. These alterations created our experimental conditions. Each treatment had a total of 3 trials, and each trial recorded the absorbance every 20 seconds for 2 minutes. Once all trials and treatments were completed, the average absorbance for every 20 seconds for each treatment was calculated and recorded in a table. This is also represented on the upcoming graph. So this figure shows the average absorbance of the reaction solution every 20 seconds over a time period of 2 minutes. The control group reached the highest absorbance of 0.83, and the 1.5 millimolar zinc concentration had the lowest absorbance of 0.74. Although the absorbance values over the time of 2 minutes was not drastically different than that of the control, there was a slight change in absorbance as the concentration of zinc increased. The graph shows that the higher the concentration of zinc added, the absorbance did decrease. This leads us to believe that the zinc ion inhibited the reaction between hydrogen peroxide and the catalase enzyme.
Figure 2 shows the average absorbance of each trial at 60 seconds. Superimposed on the columns are the 95% confidence intervals, which helped determine the statistical significance of the trials. The t-test yielded p-values of 0.368 for the 0.5 millimolar treatment, 0.104 for the 1.0 millimolar treatment, and 0.082 for the 1.5 millimolar treatment. All of the p-values were greater than the cutoff of p equals 0.05, meaning that even though a slight decrease in absorbance was observed, this trend is statistically insignificant and a conclusion cannot be drawn about the effect of zinc 2 plus on the activity of catalase. A first look at the data shows a slight decrease in absorbance of the reaction solution as higher concentrations of zinc were added. However, statistical analysis shows that these differences were insignificant with a 95% confidence interval, so we cannot draw any conclusions about the effect of zinc on the activity of catalase. We expected zinc to impact our catalase activity because the Dong et al. Study showed a strong inhibition when zinc was added. Our results were inconsistent with this study. This inconsistency could be explained by the fact that we used a catalase enzyme from plant cells while they used a bacterial enzyme. Our study could be improved by increasing the amount of molarities tested. We only tested 3 molarities of zinc and they were all relatively close to each other. Future experiments should increase the amount of zinc added to the solution to further examine the effects of adding zinc to the reaction.